First of all, I would like to start to highlight that mechanotransduction, the mechanical forces are concerning all known organisms, from bacteria to plants to mammals. Bacteria has to face osmotic shock, plants has to face wind, gravity, touch, wounding, and obviously mammals are exposed to a whole bunch of mechanical stresses. External stresses, sound, touch, and also internal stimuli concerning especially the kidney, for example, and the heart, obviously. So the next slide, please. The heart is one of the most challenged, most mechanically active organ, one of the most export organ, exposed to stretch, pressure, shear, osmotic shock, and wounding. What is well known from the heart is the fact that one electrical signal can lead to a contraction, a huge change in the mechanical environment of the cell. What is well known is also the fact that mechanics can trigger, electrics can control the electrical signal. And mechanics can basically control beating rates, can control excitation and contraction. The heart is highly mechanosensitive, and this is well known from more than 100 years, and mainly thanks to Commercial-Cordis case report. So basically, when a mechanical force is applied to the heart at a critical time during the heartbeat cycle and causing heart arrest. So this is well known in baseball, and there are many other examples like acute stretch leading to atrial fibrillation, stretch increasing spontaneous pacemaker activity, for example. So there is a long way to go from stimulation to effects. Please, yeah, from effects. And one of the most interesting points is the starting point. Basically, what are the mechanosensors and how do cells sense mechanical stimuli? There are three main kinds of mechanosensors: structural elements, cytoskeleton, extracellular matrix. They are known to transduce mechanical forces. Integrins, they can transmit the force from the extracellular matrix to the cytoskeleton. I will focus today on mechanosensitive channels and on mechanosensitive channels. And basically, mechanosensitive channels are transmembrane proteins that can be open when the membrane is stretched or curved, and while they are open, they can release ions. So basically, they can transduce mechanical signal into an electrochemical signal, which is the definition of mechanotransduction. So for some channels, for some mechanosensitive channels, we know that in-plant tension can active them, can also close these channels, and also curvature can activate these mechanosensitive channels. But for most of the mechanosensitive channels, we don't know really what can gate, open, or close these channels. And another model is the tethering model. The fact that subunits of mechanosensitive channels can be tethered by cytoskeleton or extracellular matrix. There are two main groups of mechanosensitive channels: cationic non-selective channels, where, for example, sodium, calcium, or potassium can go through without any selectivity from the channel. They polarize the membrane. And the other big group is the potassium selective channels, which can or hyperpolarize the membrane. Now, how to record and how to stimulate mechanosensitive channels, mainly using patch clamp technique. So patch clamp technique consists mainly in just sealing a glass capillary to the membrane. We can apply current, record a voltage, and vice versa. And for people who are interested in mechanosensitive channels, we need to apply mechanical force. So one way to do it is using the patch pipette, and through the patch pipette, apply negative pressure and basically stretch the membrane. So here you can see that the membrane is stretched and curved. Basically, this is a really good way to activate mechanosensitive channels. Here you can see the current, and without any mechanical stimulation, there is no current, no channel activity. And if you increase the stimulation, the mechanical stimulation, you can see one channel opening, one closure, opening again, and closure again. So basically, you increase the open probability, and if you stretch more, you increase more the open probability. The beauty of patch clamp technique is the fact that this is the only technique allowing us to follow in live conformational change of protein of single protein. Yeah, next one, please. So, for example, mechanosensitive channels, one of the key features of mechanosensitive channels is the fact that they are amongst fastest transducers. Here you can see the stimulation protocol, and here is the current with at least 50 channels open here. As soon as the stimulation starts, you can see activation of mechanosensitive channels within a range of milliseconds. So they are really fast, and also something really interesting is the fact that they can integrate the stimulation. Basically, here, if the mechanical stimulation remains constant, the channel start to close itself, so start to adapt, start to inactivate. And this is a very important feature. And another one on this channel, for example, even if the pressure is back to the resting pressure, channels here are still open. So they are not simple transducers, they can really integrate the stimulation. Yep. So something really interesting also is the fact that mechanosensitive channels are not only mechanosensors, they can sense many other stimuli. For example, here, track channel, which are potassium channel selective, can sense pH, external pH changes, internal pH changes, the heat, voltage. They can be opened by lipid anesthetics, can be sensitive to the cytoskeleton, and can be phosphorylated. So mechanosensitive channels can be polymodal channels and can be involved in a lot of different pathways, not only in the mechanotransduction. Yeah. So quite a lot of mechanosensitive channels were molecularly identified. The biggest family is the transient receptor potentials family, the TRP channels. There are also Derginac channels. There are potassium selective like TREC1, TREC2, and TRAC channels. And there are newly discovered channels like piezo channels. And all of these channels can exhibit some really different structures, from very simple structures, here in green transmembrane domains, and to very complicated structures like piezo with up to 32 to 35 transmembrane domains. So very complicated structure. Maybe here something interesting to investigate is the fact that they could sense, thanks to this structure, curvature, membrane curvature. Yeah. So interestingly, mechanosensitive channels are involved in heart disease. If TRP channels C3 and C1 are overexpressed, they can promote hypertrophic signaling, leading to cardiac hypertrophy. And in the other way around, if TRPC6 is overexpressed, it can reduce hypertrophic signaling. Interestingly, mechanosensitive channels are also overexpressed during cardiac diseases. Cardiac hypertrophy leads to TRPC1 and M4 increase, and during pressure overload, heart failure, and myocardial infarction, a lot of TRP channels are overexpressed. They are playing a major role in all of these diseases. So now, to investigate the role of these proteins in vivo, there are many tools, and among them, there is the mechanopharmacology. So basically, some drugs that can inhibit or activate mechanosensitive channels. Here, cationic non-selective channels can be inhibited by a peptide from a tarantula. This peptide is named GSMTX4 and can inhibit TRPC5, 6, and piezo, and other drugs can either inhibit trip channels or activate trip channels. And concerning potassium channels, there are also some well-known inhibitors containing TRK channels and KATP, and some activators only for TRK1 and TRK, but some really useful tools to investigate in vivo function. And a lot of work has been done in this direction using the peptide from the tarantula to inhibit cationic non-selective channels, and to show that if those channels presumably are inhibited, it can prevent or terminate atrial fibrillation. If TRPM4 is inhibited, it suppresses hypoxic reperfusion-induced arrhythmias. And the other way around, if we play a bit with TRPM4 with mutations or overexpression, it can increase or sustain arrhythmias. Potassium channels have a role to play as well. KATP inhibition leads to a block of the ischemic preconditioning. So a lot of different functions for these channels in cardiac arrhythmias and diseases. So the kind of take-home message is the fact that mechanical forces can strongly modulate heart functions. Mechanosensitive channels are key players, and they are expected to be therapeutic targets. And a very important point is the fact that myocytes are mechanosensitive, but not only myocytes. Connective tissue is also mechanosensitive. And so if non-myocytes are mechanosensitive, we can ask, what about valves? And if I can get the next one, please. What about valves? So as you all know, valves are highly exposed to mechanical forces, from planar stretch when they are closed to buckling when they open, pressure, shear, wounding. And as we have seen this morning, and we will see during the next talks, mechanical forces can modulate collagen production, ECM architecture, and stiffness. Calcification can be also modulated. And concerning Vic cells, mechanical forces can differentiate from those cells from fibroblast-like phenotype to myofibroblast phenotype. So heart cells, heart are mechanosensitive, and the obvious question is, do valves have mechanosensors and mechanosensitive channels in this case? So we've used interstitial cells from human aortic valve in primary culture, and using those cells and using patch clamp technique, what we have seen, nothing in the resting condition. But as soon as we've started to apply mechanical stress, we've seen some mechanosensitive channel activation. And basically here you can see that the more you stretch, the more you can activate the channels. And with a half activation at -40 millimeters of mercury, what we can say from this recording is the fact that in this case, we can record mechanosensitive channels non-selective for cations. We can see some nice single channel activity and this allows us to assess the size of the pore, how much ions can go through the channels, and these channels has a conductance of about 40 picosiemens, which is quite big, and we can expect from that kind of important function in valve cells. And so this recording was done in cell attach mode, basically the more physiological configuration. The pipette is just sealed on the cell, the channel is facing the cytosol. And what we can do using patch clamp technique is to excise this patch of membrane and basically get rid of soluble partners, and also to disturb cytoskeleton to assess the effect of the cytoskeleton. And what we can see is the fact that even if we get rid of ATP soluble partners and if we disturb the cytoskeleton, the activity is still there. So next, we wanted to have a look to channel kinetics and to apply cyclic stretch to mimic physiologic condition. And what we can see when we repeat our stimulation protocol, yeah, next one please. from the first stretch, we can see quite a huge current here. And if we just apply again the stimulation, the same stimulation, so please, sorry, go back again. And yeah, back again, please. Yeah, we can see that after a few stretches here, after 5 stretches, the current is much smaller. And after 11 stretches, we have almost few channels open. So we can see a huge rundown of the channel activity. So this raises quite a lot of questions. What does happen in vivo? And the valves are stretched for every second or even less. So is the channel still activated? Is the channel desensitized? A lot of questions, basically, from this experiment. So just to conclude now, I hope that I've convinced you that there are mechanosensitive channels in the heart valves. Also, I think that we can learn a lot from what we know from heart mechanobiology, and this will help a lot to understand a bit more of valve mechanotransduction. So now I have more questions than answers from this preliminary results. Sorry. The identity of these mechanosensitive channels that we don't know, and we will investigate the functions and the regulation, and obviously the role of these channels in valve disease. So yes, next one, please. I would like to now thank the organizer for giving me the opportunity to present at this very nice meeting. This was a pleasure and an honor. And I would like to thank Peter Cole team. I'm part of this team, and I also would like to thank Adrian Chester's team. All people here helped me a lot, and for presenting and getting the results you have shown today. Thank you very much, and sorry for all the technical problems. Yeah.
Questions? Yes.
Is this on? Can you hear me? Is this switched on?
Yeah.
Okay, great. You mentioned a lot of stretch work there for obvious reasons, and stretch ion channels. But the endothelial cell is subjected to complex shear stresses, which is a different, maybe a little bit different. In fact, we published the first paper, I believe, on inward rectification of K channels with whole cell clamping in the Nature paper in 1988. And that followed it up with cloning of KAR2 family and showed that 2.2 was, and maybe 2.1 are the dominant hyperpolarizing channels in response to shear stress. But I wondered whether, in addition to, in the context of your talk in the valve, you can't really ignore the endothelial cell, particularly in terms of the hyperpolarization relationship to nitric oxide, and the possibility also of gap junctional connection to interstitial cells, which would communicate hyperpolarization very quickly as a function of the environment. And I wondered whether you'd given any thought to the endothelial ion channels in terms of the valve behavior, or whether you feel it's not significant in this particular case.
Yeah, thank you for this question. Yeah, absolutely. I'm not saying that the channel I'm recording is the only one and the only mechanosensors in valve cells. Obviously, I think, yes, gap junctions can play a huge role. Voltage gated channels can also be very important. It was shown previously and on other cell types. I'm just saying that this channel is there, and we had a look on endothelial cells, and the channel is even is also present in these cell types, and we can see even more channels on endothelial cells. So it's not typical from big cells, it's also true for endothelial cells. Yep.
Yeah, thank you for your talk. Have you ever looked at strategies to overcome adaptation by, say, incrementally increasing the degree of stretch in between each cycle, or making a longer rest period in between the stretching to give the system a chance to reset itself?
Yeah. Basically, I'm just starting with this kinetics, and if we wait more, we can get more current. So basically, if the stimulation is high enough, we can almost get rid of all channel activity, but the channels are still there, and if we wait long enough, we can recover the activity. This is really important, I think, regarding the fact that frequency is highly relevant for valves or the whole heart. So it might be a filter of frequencies, but this, at the moment, I can't prove anything, but this will be very interesting to follow up, yeah.
Any other questions? Okay, if not, we'll move on. Thank you very much.